All right, so we're going to continue on with Unit 3, learning targets 5 through 7, which is going to deal with the cell membrane and membrane transport. And we'll talk about the endomembrane system. We'll talk about the phospholipid bilayer, the cell membrane, and also cell walls. So cell walls are structures that give cells protection and support, and also gives them shape. And of course, animal cells do not have cell walls. Plant cells have cell walls. There's a few bacteria, but very, very small groups. So plant cell walls are made from cellulose, which is a complex carbohydrate, and we talked about that when we were talking about the macromolecules unit in Unit 2, learning target 9. So fungi and bacterial cell walls, both of these have cell walls as well. As I said, there's a few bacteria that also have cell walls. They're composed of different material and they serve additional functions. So with bacteria, the cell wall will give it the gram-positive, gram-negative characteristics, which we won't talk about much in this class. But if you move on to upper level biology classes or move into veterinarian science classes, and you definitely will talk about the gram-positive, gram-negative characteristics. So the plasma membrane or the cell membrane, again, is composed of a phospholipid bilayer. Bi meaning two, so there's two layers of phospholipids, and we talked about phospholipids as well in the macromolecules unit when we talked about lipids or fats. So keep in mind that phospholipids are composed of two parts, a phosphate group and two hydrophobic fatty acid chains. And remember what hydrophobic means. It means water fearing or scared of water. So the inner layer of the bilayer is hydrophobic or water fearing. So, and I know my bar is popping up here, but where the tails are pointing at each other, that is a water fearing area. And the outer layer is hydrophilic, which means water loving. Semi-permeable membrane. So the bilayer provides a cell with protection and allows the membrane to be semi-permeable. What this means is some materials can pass through the cell without any assistance, pass through the cell membrane without any assistance, and some are blocked, typically based off size or maybe ports, so to speak. Um, so the membrane utilizes surface proteins in order to allow the passage of some materials. The phospholipids, along with the membrane proteins and other molecules, is called the fluid mosaic model. And again, I should have said this earlier, feel free to pause this video and get these notes whenever you need to, and then continue to push play. So the fluid mosaic model is the idea that the elements of the cell membrane exist together fluidly, and what that means is they're constantly moving. The cell membrane is not rigid; it's not static. It's always moving. The protein channels and different types of ports are continuously moving around this phospholipid bilayer, like liquid, like water, just the things in water floating around, moving around. All right, so again, solutions. So solutions are a mixture in which the solute has completely dissolved in the solvent. In cellular life, the solvent is almost always water. And this is going to be important when we talk about concentration gradients here in just a minute. So what is concentration? Well, concentration is the amount of a solute per volume of the solution. And you can see here's the equation, mass of the solute divided by the volume of the solution. So the big thing is concentration gradient. So this is when the concentration changes from one area to the next. So if we're talking about the cell membrane, then let's say maybe outside the cell you have a higher concentration of sodium ions than you have inside the cell. So you have a concentration gradient on both sides of that membrane, okay? Now, keep this in mind. Nature naturally wants to move to equilibrium. And what equilibrium is is that it's equal concentration on both sides of some sort of barrier. Okay, for what we're talking about now is the cell membrane. So naturally, if you have a higher amount of sodium, and it could be whatever, um, outside of the cell, it's going to want to move inside the cell to even itself out. Okay, nature does not like concentration gradients. Now, the great thing about this is it provides energy, okay, once those particles are moving across, which allows the cell to do work. And that's basically what this whole slide is saying. There's also a good example here of the bicycle. Okay, you can be sure to look at that. Um, and so if you're at the top of the hill and you're riding down the hill, that's how nature works. That's naturally what's going to happen. But if you got to get to the top of the hill, think about going against the concentration gradient. A lot of energy has to be put into it. So if we were trying to pump more sodium ions out of the inside of the cell to the outside of the cell, and the outside of the cell has a higher concentration, then we have to do active transport. We have to put energy into moving those materials because naturally it does not want to do that. So passive transport is a transport of materials across a cell membrane that requires no energy. Typically, this is done by the concentration gradient. If we have a higher concentration outside of the cell than inside of the cell, then it's going to passively move itself through the cell membrane if it's allowed without the use of any energy. And the opposite is true. If you have a higher concentration inside of the cell than outside of the cell, then naturally it's going to want to move outside of the cell. So no energy is used here, but energy is produced. All right, so one of the ways is diffusion. So this is the movement of materials from an area of high concentration to an area of low concentration. The diffusion of materials across the membrane releases energy. So this creates a potential for the cell to do work. So again, the definition of equilibrium is when the concentration is equal on both sides of the membrane. Osmosis. So this is the diffusion of water across a semi-permeable membrane. So diffusion is the movement of particles. Osmosis is the diffusion of water. Okay. Um, the same idea here. If you have more water outside the cell than inside the cell, then water is going to move into the cell, and vice versa. Osmotic pressure. This is the pressure acting on the membrane as water diffuses from an area of high concentration to an area of low concentration. And there's three important vocab terms here: hypotonic, hypertonic, and isotonic. So hypertonic, the side that has more solute than water. Hypotonic is the side that has more water than solute. Isotonic is when both sides have an equal concentration of water. And that's important for you to understand. It's important for you to have these vocab terms down uh, really well. So facilitated diffusion is a diffusion through the assistance of a protein channel. Okay, that's what we're seeing right here. Specialized or specially designed protein channels that allow certain materials, specific materials through the cell membrane. All right, so active transport is the transport of materials that requires energy. This type of transport is across the concentration gradient. Usually needs the assistance of a pump, a protein that actively pushes the materials across the membrane against the gradient. So if we look at this picture, we have a higher concentration of protons outside of the cell than we do inside of the cell, but the cell's wanting to pump those protons out. Okay, so we're going against the concentration gradient. We're going against what nature wants to do. So energy has to be used to actively move those particles against that concentration gradient. All right, last two slides will be endocytosis and exocytosis. So endocytosis is a type of active transport that involves the passage of large materials into the cell by pinching off part of the membrane that surrounds the materials. Phagocytosis is when it takes in solid particles in this process, and pinocytosis is when it takes in liquid particles using this process. So the cell membrane will literally pinch itself off to bring in whatever, whether it be solid or liquids, and create a vesicle, which is it's a phospholipid bilayer, little vesicle is made of the same thing that the cell membrane is made of. So, endocytosis, bringing things inside the cell. The opposite is exocytosis, which is the opposite of endocytosis. Particles exit the cell, and the vesicles containing the material become part of the cell membrane. So while endocytosis is creating those vesicles and pinching them off and sending them into the cell, when the cell releases waste or other type of material, maybe proteins that are made, those are released in vesicles as well, and they go to the cell membrane and they fuse with the cell membrane and open up, and then they become part of the cell membrane. So the cell membrane is continually getting replenished even when endocytosis is using some of it up. And that is the end of learning targets 5 through 7. If you have questions, let me know. Be sure you complete the video form, and we will continue on next week. Thank you.